very soon. Amazing. I think we're live. So welcome everybody who's tuned in via YouTube Live. We're here for another session of i3. We do this every Wednesday, 1st and 3rd of the month. So this is, of course, the 3rd Wednesday of August. So the next one will be the 1st Wednesday of September. So do tune in again for that one. And we have two really exciting talks for you this week. We have Lilian Kissiswa, who's coming from the Danish Research Institute of Translational Neuroscience, which will be really exciting. And later on, Juan Pereiro from the School of Physics in Cardiff University. So we're going to start with Lilian. Really exciting talk coming up. So I'll let you introduce yourself, Lilian, and
Okay.
looking forward to it.
Okay, thank you very much. Let me just share my screen with you guys. Can you see now my screen?
Yep, perfect.
Brilliant. So thank you very much, and thank you for the organizer for inviting me to give this talk, and of course for everybody who's tuning in to listen to us. So my name is Lilian Kissiswa. I'm an assistant professor at the Danish Research Institute of Translational Neuroscience. I belong to the Department of Biomedicine here at Aarhus University in Denmark. So what I would like to talk to you today is about aging and how can we combat this aging, or at least how are we thinking as researchers, how we can combat this. So before I go deep into the talk, I would like to just clarify two terminologies that I'll be using throughout the talk. And the first one is lifespan. I assume most of you know what lifespan is, but just to be on the safe side, basically what it means is the length of the time from the time you're born to the time you stop existing, basically until you die. Now, the other term that I'll be using here is a health span. And what we mean with this is basically the health life expectancy. So basically the time during your lifetime where you're completely healthy and you're not having any problems or disabilities. So it's common knowledge now that life expectancy has increased over the years, and we can see this trend basically starting already from the time they started recording this in the 1700s. And what we see is that, for example, we live much longer than our forefathers used to live. If you look at back in the 18th century, in the 1800s, where people lived up to only 30 years, sometimes 40 years, we have actually doubled that, and we live longer than 80 years. Actually, in Europe, currently is that we are living at the average life expectancy is 81 years. This is not a luxury that is only restricted to the Western world. Other parts of the world also have seen this increase in life expectancy. So this is fantastic, but does that actually mean we live a healthy, long life? The answer to that is no. So unfortunately, if you look at the statistics, and these are from the European Union, we can see that the average time of anybody in Europe that live a healthy life is only 64 years. And some countries obviously have a little bit longer. For example, in Sweden, where they live up to 73 years, they have a health span of 73 years, and you have some other countries that are a bit lower than that. But generally, within the European Union, the health span is about 64 years. So this is a bit worrisome because obviously it does not match up with the lifespan that we have currently. So as I said, we are living over 80 years, and the health span is only 64 years. So what this means is basically 20% of your life, we are living in a very bad health, very bad functionality, productivity, and depending on other people, either partially or completely. So that what this amounts to is about 17 years in average for a European person that they live in bad health. And what happens at this point is basically we start getting age-related diseases. So age is the biggest, is the predominantly risk factor for age-associated chronic diseases. And I have put I have shown here the different kind of age-related chronic diseases that we have currently in the world. And this varies anything from neurological diseases, metabolic, cardiovascular, chronic inflammatory diseases, and many more. What I would like you to take from here is that the diseases are not restricted to specific parts of the body. It's basically every single part of our body is subjected to age-related chronic diseases. So the problem is now that we get these diseases, and so now we have our elders that are living longer, but they're also sicker longer. And in fact, the saddest bit is that a majority of these older people, they are not only fighting one disease, but sometimes they can be coping with multiple of these diseases simultaneously. So this is obviously very bad for the individual affected, but it also affects the lives of the families, the caregivers, and also society basically in general is also affected. The statistics doesn't get better, unfortunately. What we can see is that age-related chronic diseases constitute the majority of disease burden in the world. And to make the matters worse, they're also the most common cause of mortality in the world. And here are just some statistics for specifically for United Kingdom, but this is a trend that we see everywhere in the world currently. So a lot of good and tremendous research and also advancement have been done to study these diseases and try to better understand the cause of diseases and even to develop treatments for these diseases. And they actually, some of the treatments we have, they do work very well. The problem is they work to decrease mortality, but they do not prevent or reverse the overall decline of health within the society. And the other thing that we have been experiencing in the past few years is that once we start understanding one disease and we actually try to make a treatment for that disease, something else always pops up. So it's a matter of this sort of chase toward against all the diseases that are coming up. So what our group and also other groups in the world, especially in America, that have now trying to focus is that to propose that instead of actually going and studying every single disease and try to develop a treatment for every single disease, it will be more beneficial and effective if we can actually try to study and understand aging and try to understand how it enables diseases, and in that way we can either develop a better treatment towards aging, which will lead to better treatment for diseases, but in the best case scenario, it could also lead to that we eradicate the occurrence of these diseases. So this is easily done, or easily said than done, obviously. So the question is that now, where do you start? So our lab, we thought like, okay, fine. So let's see at what point does this disease start occurring. And in general, this is some statistic from US, but it's the same in the European Union. In general, we see that this disease starts already at the age of around 60s, I mean, give and take. So what that means, or what that tells us is basically there's something that is happening before age of 60 that leads to this catastrophic outcome. So we are more interested in the neurological diseases. Obviously, there are other things, other labs that are working on other age-associated diseases, but we are focusing mainly in the neurological diseases. And here again, just to remind you that you see that the older you are, the higher chances are that you will get one of these neurological diseases, including dementia, so Alzheimer's, Parkinson, and ALS, and many other more. So what we also know that in this neurological diseases, one of the hallmark is that we have interrupted cell communication. And what I mean by that is that neurons, they have this fantastic morphology where you have the cell body and then you have the processes that are going from the cell body, which are sent out to be able to contact other cells and also other parts of the organs in the body. And in that way, they can establish communication so the information can come from one cell to another cell. But in these neurological diseases, this communication is disrupted because these processes somehow they start dying back, and then the cells cannot communicate with each other. And when the cell starts sensing that it cannot communicate with its environment, it will induce something that we call programmed cell death. So basically the cell will realize that I'm not useful, so it will kill itself. And when the cells start dying, that's when the symptoms of the neurological diseases start appearing. So we asked, does this happen in aging? Maybe it could start happening much earlier. And then what we are seeing in the neurological disease is more or less like an end point of this process. So what we have done is that to try to map the morphology of neurons at different ages. And here I'm just, to simplify things, I'm just showing you data from adults, so young adults, neurons, and then the aging neurons. And as you can see here, so we have now labeled these neurons with a fluorescent GFP, and then using microscope, you can take images to see how the morphology looks like. And I hope you can appreciate that there's so much difference between these cells in comparison to this one. This is much, much bigger in comparison to this one. We can then go in to actually be sure that this, what we see is actually what we think it is. So we can go in and start counting the number of branches of these processes and also the length of the processes. And we can quantify this, and we can see that in aged neurons, you see a decrease in branches and a decrease in length. So what that means that during the aging period, so these cells slowly start retrieving, or the processes start dying, and then they cannot communicate with each other. And then eventually you will get Alzheimer's or other neurological diseases. So the approach we are taking in the lab is to look at cytokines. And the reason why we started looking at the cytokines, because we know from literature and some of the work that I did when I was in Cardiff, in Professor Alan Davis' lab, we could see that a lot of the cytokines were expressed in the brain. And these cytokines, basically, they were very important for the growth of these processes during development. Some of them they could promote the growth of the processes. Some of them had a detrimental effect of the processes. So, and this is the reason why we started looking at the cytokine. So the question we ask here is that, does the mechanism that we see during development also take place during aging? And of course, the first thing you would do is to find out if the cytokines are actually expressed in adult brain and also aging brain. And we have, to our surprise, found that there's over 23 cytokines that are expressed in the brain. We see some of them are highly expressed in aged brain in comparison to young adults. Some of them are expression has decreased. But there are lots of them being expressed. So the task we have now is to basically go in and try to see what each of these cytokines are doing, if they have any effect or any impact on neuronal morphology, and that might lead to neurological diseases eventually. So, of course, this is not undertaking that I'm doing by myself. This work involved a lot of labs and a lot of people, and I'm quite lucky to have very many collaborators, both from here at Aarhus University and in the hospital, but also from other universities in the world. For example, Karolinska Institute, which is in Sweden, the University of Singapore, Poland, and also the Max Delbruck Center in Berlin, in Germany. So all these people are somehow involved in the project, either by providing reagents or even brainstorming and those kind of things. And I'm very grateful for my collaborators. I would also like to acknowledge, obviously, the institutes that are hosting my research currently. So the DANDRE, the Danish Research in Translational Neuroscience, and the Center for Protein and Memory, and obviously Aarhus University, and obviously the funding agencies. I would like to finish off here with saying thank you for listening and participating. And if there's any question, I'm quite happy to take them now.
Woohoo! Thank you very much, Lillian. That was a really, really interesting talk. Hopefully one day you can stop us all growing old effectively. So obviously we're streaming this via YouTube Live. So if you have any questions, please type them in the comments. There's a live chat thing I believe to the right of the window. There is a short delay. So whilst we wait for any of those to come through, I'll start with one of my own questions. So I was wondering, once you actually identify these cytokines that can be implicated in aging, how is it that you can possibly use that to either prevent or slow this process?
So yeah, that's a very good question. So basically, first of all, we need to understand if they are implicated or not. And if they are implicated, there are other ways we can either use a pharmacological approach where we basically develop a drug that can either enhance the effect of the function of the cytokines or decrease the function of the cytokines. That's one way to go. But we also do know that dietary can also affect how the cytokines works. So basically what people have been suggesting that there are some bioactive compounds in things like vegetables and spices and fruits that could also have an impact on aging and actually have a beneficial impact on aging. So it's not only that of course, it would be good if we can get a drug that can use it, but it's not only the drug, there's other avenues that we can actually follow. But before we do that, we need to understand the mechanism first.
Amazing, thank you. We've had another question come through on the YouTube from Borja Lagonja, and he said, "Great talk, thanks. Has your lab looked at the impact of telomere shortening in aging and disease? Is it a factor?"
It has. We haven't looked at it, but it has been implicated in the aging, obviously. And the thing is that it is, in some cases, it has a very big impact. In certain cases, it doesn't have. So I cannot really say for sure if that's the only thing. I don't think it's the only thing. Aging is a very complex process. So I think it would be a combination of a lot of things. It could be cytokines, it could be the telomeres, it could be anything. So we have to look at it more in a systematic manner. So no, but we haven't looked at it ourselves.
Amazing, thank you. And another question from Bhavna Gupta. How possible will it be to bridge the gap between lifespans and health span so they basically coincide?
Yeah, that is the $1 million question. And I would like to tell you that, we can do this now. It's possible. I believe nothing is impossible, actually, to be honest. But the thing is that it will be a combination not only of our scientists trying to find out the mechanism, but also a combination of living healthy life. Try to eat a good diet and exercise as much as you can if you can do that. So I do have a feeling. If you look, for example, in Scandinavia, in Sweden, people live up to the health span is about 73. But if you look to other places, like let's say countries like in Latvia, which is only 51 years. So that means that there is something in Scandinavia that people are doing that actually increasing this health span. So it's basically much closer to the 80 years of lifespan. So I think it's not only a matter of waiting for the scientists to find a cure, but I think we as ourselves, we can also increase this by basically living very healthy.
Amazing, thank you. And we've had Sri Panaswamy come through as well, saying, "Good talk, Lillian. Thanks for accepting to talk."
Thank you.
I'd like to echo that. Thank you so much. It was a really, really interesting topic and an incredible talk. If anyone else has any questions for Lillian, please do feel free to put them in the chat, and we can always come back to them at the end. But until then, thanks so much for talking for us.
Thank you very much for inviting me.
No problem at all. It's our pleasure. But in the meantime, we'll move on to Juan Pereiro, and he'll be talking to you about growing crystals to create light. So yeah, take it away, Juan.
Thank you. So let's start. Thank you. Thank you very much. So thank you very much for inviting me. So I'm Juan Pereiro. I work in Cardiff University in the School of Physics and Astronomy. I'm going to talk about growing crystals to make light, which is in the framework of electronics. So if you think about in the last 10, 15 years or so, life has changed quite a lot, right? We are connected to the internet, and it has changed on how we communicate. A lot of us are constantly looking at the phone or at the laptop or at the tablet. And all these changes have been supported by the change in electronics that we have had around us, right? And the power of electronics around us. And in the next 10 to 20 years, we expect things to keep on changing, right? We expect the Internet of Things to develop. The Internet of Things is that situation where things can connect to each other without needing us in order to connect them. So for example, our fridge can order some food for us if we are running out. The cars will be able to communicate with each other, so they can drive themselves. There will be more efficient materials to generate energy. There will be different ways to carry out healthcare, for example, to be able to do it in the distance, more robotics, more artificial intelligence around us. But in order to do that, we need electronics to keep on developing. Currently, electronics are based on mainly one single element, that is silicon. Silicon, we use it because it allows us to control whether the material conducts electricity or it doesn't conduct electricity. It conducts electricity. If we choose for it to conduct electricity, we generate this 1, and if we choose it for it not to conduct electricity, we'll generate a 0, and we process information in this series of 1 and 0s that are done with very, very small switches that we call transistors. So far, the development of electronics is following what is called the Moore's law, which was something that Gordon Moore, which was the co-founder of Intel and Fairchild Semiconductors, said in 1965. And he said that he expected the number of transistors of these small switches in chips, in integrated circuits, to double every year. And he said this with respect to 10, 15 years, but it's something that it has kept until effectively today for 45 years or so. Well, actually 55 years or so. And you can see in the graph actually that the progression has been quite constant, isn't it? And it is now that we are seeing that there is some slowing down of the progression, and this is because we are making these transistors, these small switches, so small that we are reaching the size of an atom, and we need a few atoms at least in order to fabricate new devices. So if these traditional electronics are saturated, what are we going to do in order to keep on developing the new technologies and so on that we need? So we plan to use light, right? Light is faster than electricity, and it is more efficient. If you think about a bulb, and a light bulb, if you put your hand around the light bulb, you feel the heat, but if you try to touch the filament, you are going to end up in A&E. So the light generates less heat when it moves around the medium. And how do we generate the light? Well, there are some crystals that can generate light, right? Silicon, well, first, what is a crystal? Because I go through all this terminology, but what is a crystal? Well, a crystal is a material, right? You see here a rock, and that is silicon, and that is a crystal. What it means is that inside that material, the atoms are arranged in a periodical fashion, in that geometric fashion that you see on the corner here. And if you were to look in really, really, really high magnification to this rock, you would see this pattern repeated over and over and over again. inside the material. So a crystal is a material that has this kind of situation where you have a pattern that repeats over and over and over again. So if the crystal is right, if it has the right atoms and it has the right arrangement, it can generate light. Unfortunately, we have made all our electronics in silicon, and unfortunately, silicon doesn't make light. But we can use other materials, like the one that I have next to the silicon, it's also a rock, is gallium arsenide. It's a 3,5 material, and I will explain that in a second. In the gallium arsenide actually can create light. So we expect to use these kind of materials in order to make electronics. First, what are these materials? What are these 3,5s or gallium arsenide I have mentioned? Well, 3,5s mean that the material is made of two elements, one from the group 3 of the periodic table, one of the group 5 of the periodic table. The group 3 of the periodic table means that they have that the atoms have 3 electrons in the outermost regions of the atom, and they can share them effectively. And the group 5 means that the atoms have 5 electrons in the outermost regions of the atoms, and they can share them. So those are the electrons that generate the electricity that runs around this material. The materials are semiconductors. That means that we can control whether they conduct or not. And they are compound semiconductors, but because they are formed by 2 elements. And if you are around Cardiff, you may have heard about compound semiconductors because there's massive investment from university and industry in order to develop now the industry of compound semiconductors around South Wales. So we have 3,5s. I explained a little bit these 3,5s. As you see on the right now of the slide, you have the schematics of what is a transistor, those small switches that I was talking about. And you can see lots of different layers of different materials. All these layers are different crystals with different properties. And we need to grow these kind of materials in order to be able to make electronics. But I mean, all these layers are really, really thin, really small, right? So a few hundreds of atoms. And how do we make them? So that's effectively what I was, what I have worked on my career effectively, which is what is called epitaxy. Epitaxy is the term that refers to the growth of very thin layers of crystals. And the way it works is we have a crystal that we call substrate, that is a crystal that has been pre-made. And we put it in a chamber where we have made vacuum. That means that we effectively empty it from any gas or any particle that can be inside to the point that we are closer to the vacuum in outer space than anything else. It's fairly close to the vacuum in outer space. And then we put this substrate there, we heat it up to 500 to 900 degrees, and we evaporate the materials that we want, the elements that we want to form the crystal with, right? So this video is made with lentils, so it's just to illustrate the process. So we evaporate, and if we want to grow gallium arsenide, we will evaporate gallium arsenide. The atoms will arrive on the surface, and the atoms will move around, will find their position. Some will go, some will not, but they will form new layers of the crystals. So you see one going there. If you are going to grow gallium arsenide, then you will have to use gallium and arsenide. And the atoms really prefer to be in a situation where they are close to the crystal in the substrate and close to the crystal that they like to form. So if you have gallium and arsenic, they will try to form gallium arsenide as long as the substrate is suitable. And these are lentils, but we can actually go a little bit further and make more complex simulations in order to actually see what is going on. So this was done in the Paul Drude Institute by Vladimir Kaganer and Roman Sayduk. And you can see the atoms arriving to a surface, they form some islands, and then let's see if I get the right point. So then they will start growing, the atoms will attach to the edges of the islands will grow until they form one layer, and then another layer will start growing. So that's the way we grow them. Actually, if we change a little bit the conditions and we have lower temperature, the atoms will not move around that much, and they will grow these kind of mounds and rough surface that we may want to grow depending on what we want to do. Effectively, we cannot manipulate the atoms, but we can control how things grow depending on the conditions that we choose to work. But the understanding of how the crystals form is very important, and that's what I do. And now, unfortunately, I'm going to go a little bit more technical because this is what I do. But hopefully, it's only a few minutes. So this is a low energy electron microscope. This is the system that I work with. Effectively, what it does is to use electrons in order to help us see what is happening during the growth of a crystal. It is a very unique system in the sense that, I mean, it's a unique system in the world because it's the only one that allows us to do videos effectively of the growth of these 3-5 materials. What we do is to use electrons, right, that come from here. They will go around the system, will go down. They will look at the sample. They will reflect from the sample, and then we will register them in a detector so we can make a video of what is going on the sample. We can look at the sample and we can see heights as small as 1 atom and widths as small as 10 atoms or so. And we can also see how the atoms are placed with respect to each other. This is important because when you usually do these kind of experiments, you would decide what you are going to do. You would put your substrate in your chamber, you will grow, and you will have to take it out of this very empty chamber. You will send it to someone will measure it, and that will be sent to someone else, someone will measure something else. Then you will receive all the data, you will look at all the data, decide how things have gone, and then repeat the experiment in order to improve things a little bit more. And you will optimize things in this long cycle. While in our case, we can do things all in one go. So we can grow the material and look at the sample and know what is happening and try to understand it in one single go. And just to end, I would like to show you some of the things that we have done. So you can see the lines correspond to 1 atom height steps, and the colors correspond to different arrangements of the atoms, so different orders with respect to each other. So you can see that we can see how things develop with time, how the atoms rearrange, and how the growth carries on. And then I have another video with a similar thing. The different colors correspond to different order. So we will see how when things grow, or in this case, the sample is evaporating, we can see what is going on the surface. And then we can actually use that knowledge in order to try to understand and do our own simulations and then understand what it is actually going on. So hopefully after this talk, you have got a little bit of an idea of what crystals are, how they are grown, and how we can use them for electronics, and what I do in Cardiff University. Thank you very much. I want to thank the people that have been involved in the group. So Professor David Jesson, who has recently retired, 2 PhD students that have recently graduated, Dr. Daniel Gomez Sanchez and Dr. Kenneth Rodriguez Hanikainen, and the people that still remain in lab with me, Robert Lang, Matti Ivanov, and Leo Plataler. So thank you very much. And if you also thank the funder, CPS and Marie Curie. So if you have any questions, happy to answer. Thank you.
Thank you very much, Juan. That was really interesting. So as before, because we're on YouTube Live, there is a short delay. So if you do have any questions, feel free to put them in the chat, and they'll appear just in a moment. But until then, I'll just get the ball rolling. We actually have a question from Lillian.
Yes, very good talk. Thank you very much. Very fascinating, actually. So I just want to ask, and of course, you have to remember that I'm not, physics is not my background, but so when you do this crystal building, so you have the atoms in this chamber where you can manipulate the temperature and those kind of things. So obviously you will have different arrangement and those kind of things. So do these changes in temperature and changes in arrangement, will they be building different quality of crystals or completely different kind of crystals?
Depends on the material that you are working with. So if you change the conditions, if you some materials, for example, gallium nitride can grow in a square lattice or in a hexagonal lattice. So you can grow completely different crystals. In general, if you change the conditions, you will change the quality definitely. And then in some materials, you may be able to actually change the type of crystal that you're growing. Obviously, you are not going to be able to change the elements because the elements is whatever you put in.
Yeah, thank you.
Amazing. And we have another question from Bhavna Gupta. Can epitaxy, she says, good talk first. Can epitaxy be carried out in a large scale, or is it very time consuming?
No, epitaxy is used also in industry. There are other techniques that are also used in industry, but epitaxy is used in industry. So you can grow in a fairly large surfaces, and some of the LED lights that you may be using may have been grown through MBE and epitaxy.
Amazing. We've had Borja Lagonia say, great talk, thanks. And Sri Panaswamy as well. Interesting talk, Juan. Thanks for accepting to talk. I was going to ask you actually, so obviously you mentioned at the start that this is one of the applications. So this is sort of electronics and effectively advancing that technology even further. And I was wondering how soon you foresee that this could be applied to that, and basically how sort of comparatively expensive it might be.
I mean, as I said, epitaxy is used already in many applications that you are already seeing around. It's not anything new. LEDs are based on gallium nitride, for example, the white LEDs that you see, or phosphides or arsenides. They are in the electronics that you see around. It's just the development of photonics is the part that is still a little bit, because it requires combining two different types of material, and combining two different types of material is the trick question, isn't it? Because when you have a substrate that is very different to the material that you want to grow, things can go a little bit wrong. So that's the effort that we are doing in order to try to integrate silicon electronics and the materials needed in order to generate the light.
Amazing. Thank you very much. I think that's all the questions from YouTube. So on that note, I guess I'd like to thank you both again for two incredible talks. Really, really interesting stuff. And I hope you'll join us again in the future. And next week we have, in 2 weeks' time, let me just double check. That'll be the 2nd of September, the next session. And we'll have two incredible talks again. At least one on pathogens, disease, and patient risk. So that'll be really interesting. And until then, feel free to follow us on YouTube and Twitter and everything else to keep up with any updates. And of course, everything will be on YouTube afterwards, so you can rewatch these talks to your heart's desire. And I hope everyone's had a great time.
Thank you
to our speakers.
Thank you.
Amazing. Thanks a lot. And see you later.
Bye.
Bye.